Hello, welcome to Biology, the scientific study of life. Fungi, the body structure. Some fungi exist as single cells and are known as yeast. However, most species are multicellular. The morphology of multicellular fungi enhances their ability to absorb nutrients from their surroundings. The bodies of these fungi typically form a network of tiny filaments called hyphae, singular hyphae. Hyphae are composed of tubular cell walls surrounding the plasma membrane and cytoplasm of the cells. Unlike the cellulose walls of plants, fungi cell walls contain chitin, a strong but flexible nitrogen-containing polysaccharide that is also found in the external skeletons of insects and other arthropods. Fungi hyphae form an interwoven mass called mycelium, plural mycelia, that surrounds and infiltrates the material on which the fungi feeds. The structure of mycelium maximizes the ratio of its surface area to its volume, making feeding more efficient. Just 1 centimeter to the power 3 of rich organic soil may contain as much as 1 kilogram of hyphae with a total surface area of more than 300 centimeter to the power 2 interfacing with the soil. A fungal mycelium grows rapidly as proteins and other materials synthesized by the fungi are channeled through cytoplasmic streaming to the tips of the extending hyphae. The fungi concentrates its energy and resources on adding hyphae length and thus overall absorptive surface area rather than on increasing hyphae girth. Fungi mycelia are non-motile. They cannot run, swim, or fly in search of food or mates, but the mycelium makes up for its lack of mobility by swiftly extending the tips of its hyphae into new territory. In most fungi, the hyphae are divided into cells by cross walls or septa, singular septum. Septa generally have pores large enough to allow ribosomes, mitochondria, and even nuclei to follow from cell to cell. Some fungi lack septa, known as cynostic fungi. These organisms consist of a continuous cytoplasmic mass containing hundreds or thousands of nuclei. The cynostic condition results from the repeated division of nuclei without cytoplasmic division, and classified as fungi. Molecular comparisons have since confirmed that, in fact, the two clades are not closely related. Some fungi have specialized hyphae that allow them to feed on living animals. Other species have specialized hyphae called hastoria that enable them to penetrate the tissue of their hosts. As shown here in this figure, voila. Mutually beneficial relationships between such fungi and plant roots are called mycorrhiza. Mycorrhiza means fungi roots. Mycorrhizal fungi that form mycorrhiza can deliver phosphate ions and other minerals to plants, which the plants themselves cannot acquire to their own. In exchange, the plants supply the fungi with organic nutrients. There are several different types of mycorrhizal fungi. Ectomycorrhiza fungi, from the Greek ektos, out, from sith of hyphae over the surface of a root, and also grow into the extracellular spaces of the root cortex. Notice that the endomycorrhiza fungi or mycorrhizal fungi, from the Greek into extend their hyphae through the root cell wall and into tubes formed by invagination, pushing onward, which means of the root cell membrane. Okay, fungi produce spores through sexual or asexual life cycles. Fungi propagate themselves by producing vast numbers of the spores, either sexual or asexually. For example, puffballs, the reproductive structures of the certain fungi may release trillions of spores in cloud-like bursts. Spores can be carried long distances by wind or water, and if they land in a moist place where there is food, they germinate, producing new mycelium. To appreciate how effective spores are at dispersing fungi, try leaving a slice of melon exposed to the air. Within a week or so, you should see fuzzy mycelia growing from the microscopic spores, constantly falling into the melon slice. Sexual reproduction: the nuclei of fungi, hyphae, and spores of most species are haploid, except for transient diploid stages that form during sexual life cycles. Generally, sexual reproduction in fungi begins when hyphae from two distinct mycelia release sexual signaling molecules called pheromones. If the mycelia are of different mating types, the pheromones. If the mycelia are of different mating types, the pheromones from each partner bind to receptors on the surface of the other, and the hyphae extend toward the surface of the pheromone. When the hyphae meet, they fuse. This compatibility tests contributes to genetic variation by preventing hyphae from fusing with other hyphae from the same mycelium to another genetically identical mycelium. The union of the cytoplasm of the two parent mycelia is known as plasmogamy. In many fungi, the haploid nuclei contributed to by each parent do not fuse right away. Instead, parts of the mycelia contain coexisting genetically different nuclei, such as mycelium is said to be heterokaryon, meaning different nuclei. In some species, heterokaryotic mycelia become mosaics with different nuclei restricted to different parts of the network. In other species, the different nuclei mingle and may even exchange chromosomes and genes in a process similar to crossing over. In some fungi, the haploid nuclei pair of to a cell, one from each parent. Such a mycelium is said to be dikaryotic, meaning two nuclei. As a dikaryotic mycelium grows, the two nuclei in each cell divide in tandem without fusing. How hours, days, or even centuries may pass between plasmogamy and the next stage in the sexual cycle, karyogamy. During karyogamy, the haploid nuclei contributed by the two parents fuse, producing diploid cells, zygotes, and other transient structures from during karyogamy. The only stage in which diploids exists in most fungi, meiosis restores the haploid condition, and the mycelium then produces specialized reproductive structures that produce and disperse the spores. The sexual processes of karyogamy and meiosis generate extensive genetic variation and important prerequisite for adaptive evolution. The heterokaryotic condition also offers some of the advantages of diploid in that one haploid genome may be able to compensate for harmful mutations in the other